to spend just a short little period of time talking about the requirements for mitosis, and then actually talking about mitosis itself. Now, again, in the previous video, I just discussed, the different phases of interphase, the bulk of the cell cycle. And, I discussed about how these are basically preparatory phases for the reproductive aspect of the cell, for the actual division itself and the daughter cells, right? But before the actual, mitotic events take place, the cell has to be perfectly prepared, to undergo that process. And there's a few factors that are going to influence whether or not it's even right for the cell to divide. All right. One of them is there just enough space? Is there just enough room for mitosis to take place? All right. But you have to remember, tissues are, tissues are composed of many cells, but these cells also fall within a border. All right. And these cells, not only contact themselves, they contact the borders of the cell. All right. So if there is no room for mitosis, then, due to contact inhibition, then there will be no mitosis taking place. All right. So like now, what can go wrong with this though is, again, if you want to go back to the cancer aspect of this, let's say we're talking about a benign mass. Benign mass is more a slow-growing mass of tissue. So what will happen here is, basically these cells don't abide by the rules of contact inhibition, and they just continue to divide within the borders of the cell itself. And then what will happen is that will expand the area of that tissue. So that tissue is going to kind of bulge out, all right, because cell growth is not being regulated, not controlled due to something that went wrong during interphase. All right, and the reason why we consider this benign is because, one, like I said, benign masses are typically slow growing, but another reason why is this growth stays within the borders of the tissue itself. All right, benign masses are non-invasive, and they don't actually grow out of the borders of the tissue and directly invade other tissues. All right, surrounding tissues, I should say. The danger with these, though, is that the reason why cells need to respect borders is because there's only so much room within the human body. All right, and if our tissues start to expand, beyond their normal borders, beyond their normal size, then that's going to apply pressure to the adjacent and surrounding tissues. And this can be bad, for example, if you have a benign mass growing and it starts to compress an artery. All right, if it starts to put pressure on an artery, that's going to impede blood flow to tissues that need that artery. That could kill the tissues. All right, or it could, you get the picture here. All right, so that's basically, one requirement is there needs to be space. A cell has to die somewhere in here. There has to be some kind of tissue damage, so there's room for the cells to actually, proliferate. There has to be the right, the cells have to be within the right environment. Okay, and what I mean is when the cells are in the right environment, there needs to be certain chemical factors, chemical messages that are going to stimulate this process to begin with, and that's what mitogens and growth factors are. All right, they're basically chemical messages that are secreted by surrounding cells or the tissue within that basically stimulate. Okay, look, we need cellular division to take place. All right, so then they'll like any other chemical message, they'll influence the activity of the cell, and they'll influence it in a way that basically start to turn on the mechanisms of growth, to basically take the cell out of that kind of perpetual state of gap zero and move it into interphase, into G1 synthesis and G2. All right, then which will eventually lead up to mitosis, the division itself. All right, so there has to be certain chemical factors that stimulate and influence this process. All right, cells have to be of the right size. Okay, cells have to, remember, you're taking a cell and you're taking one cell and you're splitting it into two identical copies. All right, that cell has to be big enough. Okay, so it can divide and still be and ensure that those identical copies are going to be the same size as the normal functional cells within the tissue. All right, and the cell is going to get larger. Because the DNA is replicating, the organelles are making new organelles. So as a result, the overall size of the cell is going to get bigger. And again, that's what the gap 1 and the gap 2 phases ensure. Like I said, they're basically cellular stall tactics to ensure that the cell is large enough and that the environment is also conducive to cell division. Energy. I mean, this is a high energy process. All right, there has to be enough energy involved, meaning the cell has to have the right amount of nutrients. All right, there has to be enough nutrients to sustain the work that it takes to make this process happen. All right, if the cell is starved, it won't divide. Okay, I mean, why do you think when you see someone undergoing chronic starvation or a cancer of some sort that they atrophy, their tissues start to shrink? Okay, because with starvation, you're not getting the calories in that you need, and if you're not fueling your body, you're going to start burning through your stores, and eventually you're going to start to run out. So then cells aren't going to be able to divide like they normally can. If you look at cancers, cancers reroute blood flow to themselves, through a process called angiogenesis. And if that cancerous neoplasm, that malignant cancer, starts to shunt blood away from your normal healthy working tissues into itself, it's literally stealing the energy that your tissues need to sustain their activity and stay alive. So as a result, you're not going to be able to turn over cells in your tissues. That's why, terminal cancer patients look so emaciated. I mean, that and, the process of the therapies that go with that. There's a term for that. They call it cachexia, wasting away. All right. So energy needs to be there. And also, this is the big one. The DNA needs to be completely replicated. Like I said, this S phase, the synthesis phase of interphase, is definitely the longest phase of the cell cycle. I mean, there's a lot of genetic material to replicate, and we have to make sure we do this right, okay? Because, during DNA replication, I mean, there has to be not only making new strands of DNA, there's proofreading involved in this, all right? So we need to ensure that the genetic replication is taking place, because remember, DNA is basically the code that drives protein synthesis. All right, we not only have to replicate all the DNA of that cell, so the two daughter cells will have the exact genetic material they need to crank out proteins and stay alive, but at the same time, we need to make sure we proofread all this genetic material to ensure that we're not making bad copies, okay? Because if this cell divides and there's mutated DNA in there, that could cause cancer. That could make a bad cell. All right. So DNA needs to be fully replicated as well and replicated right in order for cell division to take place. And like I said, if any of these factors are taken away or something goes wrong with these, then like I said, there are certain regulatory switches that basically will make the cell cycle shut down, and it will force the cell to go into what we call gap zero. Like I said, kind of a hibernation of cellular activity until whatever the problem is fixed. Sometimes it doesn't always get fixed, and the cell undergoes a process called apoptosis. Second P is silent. Okay, but basically that's programmed cell suicide. All right, so if any of these requirements aren't met and there's enough cellular damage and the cell just is not going to repair itself, it may stay in gap zero for a long time, or it may get rid of itself, programmed cell suicide. All right, and again, that's a topic that definitely would warrant a lot more explanation than what I have time for in this video. But understand that these are the requirements necessary for mitosis to take place, for cell division to take place. Like I said, that's what interphase is all about, making sure that all of this is in place. So once we meet all these requirements, once we get to G2, and then we can switch over to mitosis, then the cell is fully prepared to divide. It has space. There are the right growth factors, the chemical messages to stimulate to ensure this process is happening. The cell is big enough, there's enough energy, and DNA is completely replicated with no errors.